Hey guys, welcome back to Unstop Biology. So in today's video, we are going to understand what exactly are plant hormones, right? So I hope you remember in the last video, we actually discussed what exactly are plant growth regulators. PGRs, and what are different types of PGRs actually, and plant hormones are a type of plant growth regulators. So guys, before we start off with the topic, if you are new to the channel and you have not yet subscribed to my channel, please do subscribe to it and press the bell icon to get all the notifications. And yes, if you are able to understand the videos, please do not forget to like it and share it with all your friends. So guys, we actually understood that plant growth regulators can be promoters or inhibitors. They can inhibit or promote the plant growth, right? So basically, plant growth regulators, they can be promoters or inhibitors, right? And this applies to even plant hormones. So plant hormones or phytohormones, right? It's one and the same thing. Basically, these are plant growth regulators itself, and what we understood is that they are intercellular factors, right? They are intercellular factors, and that is why they are known as plant growth substances as well. Plant growth substances, right? So we'll start off with the first regulator, first plant hormone, which is auxin. So we'll start off with the first one, which is the group of auxins, right? So basically, there are five categories of PGRs which we are going to understand. Five categories of plant hormones, basically, which we are going to understand. The first category is of auxins. Now, auxin, the meaning of auxin is to grow, right? So this is a Greek word and this means to grow. And this was first isolated from human urine. Okay, so basically, the term auxin is for this term auxin is for indole acetic acid. Indole 3 acetic acid, in short form IAA, right? And apart from this, to other natural and synthetic compounds having certain growth regulating properties. So basically, they are generally produced by growing apices of the stem. So apices, apex or apices of stem and roots, right? And from where they migrate to the region of their action as well. So basically, they are produced in the apex of stem and root, and then they migrate from that particular apex to the region where they are required. Now auxins like say indole acetic acid or indole butyric acid. This is also one kind of auxin, indole butyric acid, IBA, right? These have been isolated from plants as well, right? And these are natural auxins. Apart from that, if you have heard of NAA, this is naphthalene acetic acid, or 2,4-D. This is basically 2,4-dichlorophenoxy acetic acid. Okay, 2,4-D. These are synthetic auxins. These are synthetic auxins. These are natural auxins. These are basically isolated from plants. Now, all of these auxins have been extensively used in agricultural practices, guys. And what they do exactly is they help to initiate rooting in stem cuttings, right? So, we usually basically use stem cutting as plant propagation, right? So, they initiate rooting in those stems, right? Apart from that, they also promote flowering. Auxins promote flowering, for example, in pineapple. Apart from that, they also help to prevent fruit or leaf drop at early stages. But they also promote the abscission of old mature leaves. So the mature leaves basically are gone now, and the young leaves and fruits are protected from because of them, right? They help to prevent them from dropping. Now, apart from that, in most higher plants, the growing apical bud inhibits the growth of lateral bud. This is known as apical dominance. So, the apical bud is always dominant, right? Now, removal of shoot tips, which is known as decapitation, usually results in the growth of lateral buds. So this also happens because of auxins, right? So basically, if you have apical buds and if you have lateral buds, what happens is the apical buds inhibits the growth of lateral buds and the apical bud starts growing. Now, if you remove that, basically the tip, shoot tip, that tip of the apical bud, basically what will happen is the lateral buds will it'll basically start growing. Now, apart from that, auxin also induce parthenocarpy. It is also known for inducing parthenocarpy, and this happens in tomatoes. So basically, they also induce fertilized without fertilization, development into fruit without fertilization, right? Now, they are also widely used as herbicides, right? So basically, 2,4-D is used to kill dicot weeds and they do not affect mature monocot plants. Apart from that, it is also used to prepare weed-free lawns for gardeners. And last thing is, auxin controls xylem differentiation and it helps in cell division. So this helps in cell division and it controls differentiation of xylem cells, xylem differentiation. So this is all about auxins, guys. Then the next category is of gibberellins, right? The next that we have to study is of Gibberellins. This is the second category. Now, these are again another kind of promotory PGR, right? So I said there is two different functions of plant growth regulators, promoter and inhibitor. Now again, gibberellins are promoters. They are promoters, right? They promote plant growth. Now there are more than 100 gibberellins which are reported from different type of organisms like fungi and higher plants. So they are basically denoted as GA1, GA2, GA3 and so on, right? However, GA3, which is known as gibberellic acid, was the first gibberellin to be discovered. So GA3 was the first one to be discovered. Okay, and basically, since it was the first one to be discovered, it was most intensively studied by various people. Now, all GAs are acids. Gibberellic acid, name itself suggests that these are acidic, right? So they are acidic. Now, they again produce a wide range of physiological responses in plants. Their ability to cause an increase in length of axis is used to increase the length of grape stalks, right? Gibberellins cause fruits like apple to elongate and improve its shape. They also delay senescence. Thus, the fruits can be left on the tree longer so as to extend the market period. So basically, GA3 is used to speed up the malting process. In brewing industry as well. Now, apart from that, you guys must have seen sugar cane. So basically, sugar cane stores carbohydrate as sugar in their stems. Now, if we spray sugarcane crop with gibberellins, it increases the length of the stem because we said that it increases the length of axis, right? And that is why basically, if you use gibberellic acid on sugarcane stems, you increase the yield by 20 tons per acre. But because what you are doing is you are increasing the size of the or the length of the stem. Now, apart from that, spraying juvenile conifer plants with gibberellic acid also hastens the maturity period, and that is why you have early seed production. Early seed production. Now, gibberellins also promote internode elongation, which happens just prior to flowering in beet, in cabbages, and many other different types of plants. So, again, guys, gibberellins are what? These are promoters, these are promotory regulators. Now, the third one that we have to study is cytokinins. The third one is cytokinins, right? Now again, cytokinins have specific effect on cytokinesis. The name itself has been derived from the process cytokinesis, and this was discovered as kinetin. In the last video, we also discussed kinetin. If you have not seen that video, you can go and watch it after this video, right? So basically, this is a modified form of adenine, which is what? A nitrogen base, a purine. It is a purine, right? So basically, it was discovered as kinetin from an autoclaved herring, sperm DNA. Now, kinetin does not occur naturally in plants, guys. You'll have to remember that, right? So, search for natural substances like cytokinin like activities led to isolation of zeatin. Now, basically, zeatin from where? From corn kernels or coconut milk. Now, since the discovery of zeatin, several naturally occurring cytokinins and also synthetic compounds with cell division promoting activities have been identified, right? So basically, since it is based on cytokinesis, right? We said it is, the name itself is derived from cytokinesis, which is nothing but cell division. So basically, it again promotes cell division. This basically promotes cell division. Right? So natural cytokinins are synthesized in region of rapid cell division. For example, say the apex, the developing shoot bud, root apices, or young fruits, and it also helps to produce new leaves, chloroplast in leaves, lateral shoot growth, adventitious roots formation, and cytokinins also help in overcoming the apical dormancy. It helps in overcoming from apical dormancy. They promote nutrient mobilization as well, which again helps in delay of leaf senescence. So again, this is a type of a group of promoters. So the first three we have studied are all promoters. The next one is ethylene. Ethylene, as we studied in the last video as well, these are gaseous in nature, right? And that is why we say that ethylene is what? It is a gaseous PGR. It is a gaseous plant growth regulator. It is gaseous PGR, right? It is synthesized again in large amounts by tissues undergoing senescence. The tissues undergoing senescence and then ripening fruits, right? Now, influences of ethylene on plants include, say, horizontal growth of seedlings, swelling of the axis, or you have hook formed in the apices, right, on the apex in dicot seedlings. So basically, ethylene promotes senescence. Obviously, it will be just opposite to what we were studying before this. So this promotes senescence, right? And also abscission of plant or different organs, especially say of leaves and flowers. So ethylene is highly effective in fruit ripening, right? And because of this, what happens is basically how it happens, it enhances the respiration rate during ripening of the fruits. And this rise in rate of respiration is known as respiratory climactic. Now, ethylene breaks seed and bud dormancy as well. It initiates germination in peanut seeds. It initiates sprouting of potato tubers. So basically, ethylene promotes rapid internode or petiole elongation in rice plants. So basically, it helps leaves or upper part of the shoot to remain above water, right? Now, this also promotes root growth and root hair formation, and it helps the plant to increase their absorption area. So, it increases the absorption area as well by helping plants. Now, it is also used to initiate flowering and for synchronizing fruit set in pineapples. Apart from that, it also induces flowering in mango. Since ethylene regulators have so many physiological processes, these are the most widely, very important guys, most widely used PGR. Ethylene is the most widely used PGR in agriculture, right? And the most widely used compound as source of ethylene is ethifon. So ethifon is an aqueous solution. It is an aqueous solution, right? And it is absorbed very quickly and then it is transported within the plant and it releases ethylene very slowly. So ethifon basically hastens fruit ripening in tomatoes, apples, and it accelerates abscission in flowers and fruits. Apart from that, it also promotes female flowers in cucumber, thereby increasing the yield, right? So this is again another set of plant not growth promoter but inhibitor. Now, apart from that, the last one that we have to study, the fifth group, is abscisic acid, ABA, right? Now, guys, abscisic acid was again discovered for its role of regulating abscission and dormancy. Like like other PGRs, it also has wide range of effect on plant growth and development. It acts as a plant growth inhibitor. Now, this acts as a plant growth inhibitor and inhibitor of plant metabolism as well. So basically, this inhibits seed germination. Right? It stimulates closure of stomata and it increases the tolerance of plants to various kinds of stresses as well. And that is why it is known as the stress hormone as well, right? Apart from that, it plays important role in seed development, maturation, and dormancy. By inducing dormancy, it helps to withstand desiccation. And then there are different type of stress factors which are unfavorable for growth. Apart from that, acid ABA acts as an antagonist to gibberellic acids, right? So basically, guys, now you know that for any and every phase of growth, differentiation, or development of plants, one or the other plant growth regulators have some special role to play. These roles could be complementary or it can be antagonistic or opposite, like it happens in case of ABA and GAs. Now, these could be individualistic role, individually they are contributing to something, or in combination they are basically contributing to something which is synergistic. Similarly, there are number of events in the life of plants where more than one PGR interacts to affect an event. Say in case of dormancy in seed or buds, or even in the case of abscission and senescence, right? Even in the case of apical dormancy. Right. So basically, all of these rules of PGR is only one kind of intrinsic control. Along with this, genomic control and then extrinsic factors, which were basically the environmental factors, also affect all of the events of growth in plants. Say vernalization, flowering, dormancy, seed germination, plant movements. Everything depends not just on the role of these chemical or intercellular, but it also depends on the intracellular and then the extrinsic growth factors as well. So with this, guys, we have finished the entire topic of plant hormones or phytohormones. I hope you would have understood the basic functions and the role of all five different kind of plant hormones. So please do like the video and share it with all your friends. And if you have any doubt, please do ask your doubts. Do not hesitate. You can ask your doubt by putting it in the comment section. And yes, if you have not yet subscribed to the channel, if you are new to the channel, please do subscribe to it and press the bell icon to get all the updates. Thanks for watching this video, guys. See you in the next one. Till then, take care. Bye bye.